Great. Okay. So I want to welcome Haneef today because he's kindly agreed to speak to us and tell us a bit about his work. Haneef is the Dean in the Faculty of Science and Education and an Associate Professor in the Department of Natural and Basic Sciences at the University of Turbat in Pakistan. Him and his group are working on local manufacturing of industrial important enzymes using cell-free extracts and fermentation technology. And he recently, within the last year, I think, received a higher education award to do this research at the university. And today he'll be telling us a bit about the production of free hydroxypropanoic acid using hydrogel compartmentalized microbial cells in yeast. Hopefully I got most of that right, but I'll pass it on to Haneef, who can tell us a bit more then.
Thank you so much, Ian. Nice introduction for me. So it's me, Haneef. Currently, I'm living in Pakistan. I'm working as an associate professor and acting dean faculty of science and engineering, as Jan introduced me. My work is basically focused on enzyme production from microbial sources. I not only I work on enzyme production, but also I work on enzyme characterization and immobilization. And today presentation, I'm going to discuss one of my projects that's I done in Imperial College London. That's production of 3-hydroxypropanoic acid using hydrogel compartmentalized microbial cell. So immobilization, compartmentalization is basically my skills which I'm going to work. So me with Professor Hidalgo, we designed a project to work on microbial cell mobilization. So we use the term compartmentalization, so plasmid immobilization there. So you work with 3 years friends and get some good results. So we are still working on these results. So hopefully get some more update on that. This is the title of my presentation, which I am in front of you. So I move to next. 3-hydroxypropanoic acid is basically a 3-carbon containing acid building block. It can be a platform for synthesis of different industrial important or commercial important products. This is the building block from acrylic acid, acrylamide, malonic acid. It can be used to produce the folate 3-hydroxypropanoic acid, which is good bioplastic as well. Acrylonitrile as well, and poropylolactin and propylendol as well. So methyl acid. So this is the basic. You can see the building block, which can be directed in any synthesis industrial product, and that can be compatible environmentally as well as economically compatible for use. Currently, synthesis of 3-hydroxypropanoic acid can be done through chemical and biological way, and commercial production right now is using chemical methods for the production. But the chemical method is some source of become very expensive, giving very low yield of 3-hydroxypropanoic acids, and starting material, you can say any material which is used for the synthesis of 3-hydroxypropanoic is also costly. And one of the major drawback is environmental incompatibility of these pathways. So people or scientists are trying to move to a biological production of 3HP. And biological production, microorganism is one of the very key flair, because it's easy to grow, it can be easy to handle, and can be produced extracellular biomolecules. So mostly the scientists are trying to use microbes for 3HP production. And microbe 3HP currently is produced in 3 pathways. So one is called 3HP mediated oxidative pathways. Using glycerol is a starting material, you can say starting material, a substrate for the production. Another one is a degradative pathway, in which acrylamide is used to degrade to 3HP as well. And third one is autotrophic, that some microbes use acetyl CoA and carbon dioxide as starting material for the production of 3HP. So these are the pathways the microbes use for the production of 3HP as a intermediate or a byproduct in their biological pathways. So commercially talk about the commercial production of 3HP. So possibility is that we need to use a cheap and abundant substances and which can be efficient. Your bioprocess method should be efficient and stable, and they give high yield of productivity as well. So third one is trying it should be a continuous or reusable or demand regulations. And this is the major industrial demand. We focus on this production. So this project contain two components. One is that they're using the construction and genetically engineered mechanism. These use and metabolic pathways to engineer the microbe for the production of 3HP process. That will all be that should be a viable, commercially viable. And second way, we will try to immobilize these engineered microbes to harness their bioactivity and use it on demand production. Whenever we need to use it, then use for the production. So immobilization then not only gives you can say a biomolecule production, but it will try to be more pure from microbial culture. So you can have some sort of partially purified your PHP and mobilization production. This is the objective that we use in this project, which I carry out. I use already engineered microbes for PHP production. The protocol is very easy. Then we just mix our culture with alginate. Alginate is basically a polymer, and salt form, in sodium salt form. It's always soluble in water. But when we mix our sodium alginate solutions and with microbes to mix it, and then we drop it into a calcium chloride solution. Whenever we change the salt of alginate, then alginate become insoluble. When it become insoluble, you see that your microbes or your biomolecules going to trap on these polymers. So this and then here you see the surface morphology that how can mobilization of some morphological change on the surface of polymer. And this is the internal structure of polymer that see the microbes distributed between and that microbe distributed not only, but there's some source of growth and this within the polymers. And this is the objective that we use recycle the project, recycle the pathways, we use it and extend the project and reuse it and again. So that is some sort of you can say a continuous production what we targeted for this. This is the plan of work. This components is we use two culture. One is monoculture, which is use single pathways from PHP productions, and we also have co-culture, two culture, which is being divided to beta-alanine. Beta-alanine is a product of one strands and RLS 875 and RLS 876 going to utilize these beta-alanine to convert it into hydroxypropylenic acid. We divide the workforce between two organisms to enhance its hydroxy production. I use these both strands to immobilize and encapsulate into a alginate beads and optimize the parameter, because you know that whenever using polymers, then we need to optimize the concentration to sustain their pore size for the encapsulation of microbes. Then we need to stabilize the calcium chloride solution is very good. Gelification of alginate depends on chloride, the calcium, so we need to optimize it as well. Bead size is very necessary for us to fraction of your beads with the substrate that you're using. So whenever the size is much smaller, you can higher surface area to do better diffusion rate and production of a product. And then we finally optimize the process. We use spatial and max commodity. Spatial mean that we use a single divide micro within two beats, and mix compartmentalization. We use mix the both strain into a same beats. Solid liquid interface also being sometimes is made. You use one culture or microbial culture in a liquid solution, another is immobilized one that you act some sort of barrier to reduce the computation between the computation between the two microbes as well. So we also analyze the solid liquid interface of co-culture as well. So finally, we check its reusability of culture that the examining compartmentalized microbes and mobilized microbes can be reusable, not then finally immobilized. These are the all about the project parameters we carry out. This is our post result we obtained. We see that both the strain show the production of 3HP. That the strain co-culture gives some sort of higher productions compared uh, to to uh, to a monoculture. Then we monoculture and co-cultures is there we can see the culture is being separated, but there is both culture is being mixed in a single. So that we analyze it that we can say that production in little bit constants as well. So it's we can see that we don't depend on anything lead to solid liquid interface. So yeah, use single production culture as well. So it is free. So I miss the slide. This is a solution form. That's mean the culture is totally in a liquid form. So you can see the immobilized form and liquid form both showing the same results. So they have not been affected. It means that your mobilization didn't affect the productivity of 3HP. You can see this morphological structure of polymers. This is the surface of the alginate without compartmentalized microbes, and this is the surface of compartmentalized microbes. You see there's smooth surface, but there is a rough surface. You see the microbes is some sort of growing in this polymers. This is the internal structure of this one, that how microbes is distributed within entrance solution. Calcium, then I moved toward the calcium, sodium alginate concentration optimization. So we observed that 2%, 3% show good productivity as compared to 4 and 5%. Because when we increase the concentration, we will reduce the pore size of polymers. When the pore size has been reduced, we limit the nutrient diffusion within the polymer. So if the nutrient diffusion into the polymer is limited, then production will get reduced. So, but whenever we use 2% concentration, that will produce a fragilized beads, very unstable beads. So that means that's lateral shaking can be degrade these beads. So 3% is show more stable type of beads, and that can be usable. So we suggest in our study that 3% is very optimum from using then hypermize the calcium chloride concentration. So 2% calcium chloride concentration is very good to produce stable beads from mono and co-culture for the production of hydroxyfluoric acids. And whenever we reduce it, so we maybe affect the because it's maybe affect the bead size as well, pore size as well, and as well as more compact beads, so limit the nutrient as well. And calcium chloride itself, the concentration highest, maybe shift some source of nutrient value of microbes. So that is therefore the production being reduced or to increase of calcium chloride concentration. This is the incubation period. Fermentation period is very important. So we optimize that. That's our free culture. That's without immobilized one. That's immobilized one. We see the monoculture. That is monoculture, and free one is so higher production in 24 hours, but immobilized show higher production in 48 hours. Because when you talk about the polymers maybe limit the interaction so between nutrient and microbes. So that maybe need time for diffusion and into the polymers, and that maybe take time more time. So that is a little bit drawbacks for production industry, that we need more time for production of the hydroxypropanonic acid compared to free one. So that's mean that's the endure there becomes similar show. That's higher production we get in 44 or 48 hours. That up to that production has become constant. So ratio of salts, we use different concentration of the salts and vary the ratio of the salts between the co-culture. That's always depend when you're using two strand, then you have to optimize the ratio. So 0.5 ml micro ml ratio, use the culture ratio into the growth. So that's you give us very production of 3 HPs compared to the lower concentration differences. So that's mean or if you suggest if using using these two strands, it's and 1:1 ratio is good for them to 3 HP 3 hydroxy production. This is solid liquid interface. We use both. We change the space of micro. We use one strand is RLA 75 in liquid and RLA 876 is a solid. So that vary that between them, and then we say that we didn't see a major difference in the production. So it's up to us the which strands can be a liquid, can be a solid. If someone try to this one, so production has always been constant when you as compared to the our previous immobilized strand. This is the restarting of land. So we use same beads, and we see that almost the production is going to be constant. This after that third cycle of 7 to 9 cycle productions is being going to reduce continuously. They're using because of these fertilized system, but monoculture little bit show more stability in terms of recycling as compared to the co-culture strands itself after mobilization. So it's mean that the reduction of production is might be due to the you can say diffusion of the cell. Sometimes using cell to cell, that cell may be some sort of cause a diffusion out of the polymers. So reduce, and then we wash it. After washing, these strands may reduce, and then production may be reduced as well. So, but stability is quite good in terms of storage. If you use the leads and I store in up to 30 days of and every you can say 10 days I check the production. So almost the production of both strands in constant. So they show or very constant results as compared to resetting. So that's storage means that we are strand more stable of our encapsulation. So this can be a conclusion of my project that 3% as mobilization. If you doing a mobilization, 3% alginate, 2% calcium chloride show high production of the hydroxy propanic acid production. The microbial compartmentalized hydrogel beads with smaller bead size give higher production as compared to the hydrogel bead for higher bead size. The Saccharomyces cerevisiae transformation, basically the production time is increased from 24 hours to 48 hours after encapsulation. So co-capsulations and single bead with 1:1 ratio give 65% higher productions compared to encapsulated strands separated originates. So if you get separated gel beads, then we have a very limited production. If we change the beads in the same mixed culture will get high productions compared to separated one. The production is in continuous to 100% of the activity of 10% beach per month position, but little bit reduction of production. You see, the storage is being 100% and the rate shrinking process as well. These are some sorts of project currently we are working. I'm in my lab, so local manufacturing for commercial important design using microbial fermentation technology. Next generation biofuel production using dead form waste is for biofuel production using microbial strain as well. We also designing enzymatic biofuel cells, so that is some source of we use oxidase enzyme or oxidate redox enzyme to design some source of biofuel cells that we use for feast networks or some source of other biological based devices. Thank you so much for listening. That's all about me. Any questions now?
Thank you, Haneef. That was really interesting. I'm just going to open the floor to the audience. Does anyone have any questions? I guess whilst you guys are thinking about this, I have one quick question about these using the system, the immobilized system you have. Is it also compatible for making productions of other enzymes beyond? I'm going to go with 3HP.
Yeah. Because immobilization is some sort of just a techniques which can be a trial for any microbial strains and any biological system. The concept is something that you just want to trap, you want just to retain some source of their biological activity with to make it reusable for another process as well. So it's mean you want to make in one of our project we equally suggested we never execute the project that we as I suggested them that if you use a free cell extract for the production, then you want to immobilize these free extract into a system. So you maybe get some source of fry for these free system is a manufacturing. So it can be a try for any type of microbes, for any type of enzymes or biomolecule production. You itself use it for enzyme mobilization as well. So to design some sort of enzymatic pathways as well, which is not possible for biological systems as well.
And just following up on that, I think I may have missed it, but is it relatively affordable to be able to make this system yourself then, or do you need a bit more specialist equipment?
No, it's not need a more special equipment. If you want to characterize it, then maybe you need some sort of routine polymer characterization. But I don't think it's no, because it's just you just bound them. You just encapsulate them or bound them. If you want to try to bound them, that you just need some source of chemical bonding between your biological system to a support. You just maybe use a cross-linking agent. That's just mixing them. If you're using a polymer, then you just mix them with the polymers and then just to convert as liquid into a solid. So what we do in alginate form, alginate is always in a soluble form when we use in salt form and sodium salt form. So sodium alginate, it means it's a soluble, but whenever you convert into a sodium into a calcium alginate, it's become insoluble. It's become some sort of solid material, because because ions charge is being changed, because sodium is 1 plus and calcium will be a 2 plus. It's easily replaceable by calcium. To calcium easily replace the sodium. When sodium is some source of, then maybe you can try it as a solid material. So just you mix your biological system and convert it into a solid, then the solid material is said that, yeah, that's mean it's affect somewhere, so you can make reusable some sort.
Oh, great. Sorry, Charlie had to leave. Sorry about that. No worries. Is there anyone else? Does anyone else have any questions? Feel free to put in chat or say out loud if you want to.
I have some questions. So thank you, Haneef, for the nice talk. So for me, I only heard about enzyme immobilization. It's the first time I heard about microbial immobilization. In enzyme immobilization, the effect is the immobilization makes the enzyme more stable and reusable. In microbial cells, how does the immobilization help in the increase in the yield?
Can you kindly repeat the question so I may unable to.
Yeah. In the immobilization of the yeast, how does it? What is the mechanism by which it increases the yield as compared to the non-immobilized ones?
Yeah. That's difference is not so much because both show some sort of good results, but immobilization is something like that that's going to affect this one. So you can say that our target is some source of manufacturing because to reusable. So it doesn't affect the production. Maybe little at time it's been reduced, but it's not affect is so much. So it's little bit reduced as well. So that's the production. But overall, the concept of production is both are same. Just time. I just mentioned the time has been changed because it affects the time of manufacturing, but it doesn't like change.
So it's more of the reusability than the increase in yield.
Yeah.
So I had a person message and they were saying apologies for being naive, but what can free HP be used for in industrial purposes? Sorry, Haneef, did you get that question? Sorry, you're on mute, by the way.
Sorry, I didn't get the question.
No, they messaged me directly. They wanted to ask basically what can free HP be used to do? Like what are the chemical products? What essentially they were asking what are the industrial purposes for using free
You
HP?
mentioned earlier that 3HP is some sort of starting or building block for acrylamide, nitrile, trilic acids. So these are 1,3-propional. These are the used for the drugs in capsules. You can say surface capsule ending design. Poly 3HP is also a product of 3HP, which is used to produce bioplastic as well. So a good replacement of plastic as well. So nitrile is something that's been also used for this one. So that's generally we can see that's plastic industries and some sorts of carpeting industries, and you can see also some sorts of pampers of child as well. You can say acrylic acid is being used and these type of products. So that's these are generally you can say you will target some sort of a good replacement of industrial product as well.
Yeah, I have another comment or question. Yeah, I think wouldn't this be useful when the product is kind of toxic to the cells itself, right? Because there's a chance that you could separate the toxic product, that you can just use this on this purpose, right?
Yeah, because PHP is a non-toxic and biological-based project and biocompatible sources, so that's a good replacement of some sort of different product as well.
Oh, I
Sorry,
guess,
yeah, I
sorry,
just need my 1 minute because my son is with me. Is just
no worries.
yeah.
We welcome everyone from every age to come join us and learn a bit more about science.
So he is just yawning on me right now.
Fair enough. Well, I'll see. Is there anything? Oh, sorry. So Wendy did ask a question. She asks if this setup can be upscaled without a lot of issues. Like, how easy is it to scale it up? I guess. And again, you're on mute. Sorry.
Uh, can this is set up to be scale up without lot of issue? I don't think so because if we, I just currently you scaling up is not a big issue for such a type of project, but it depends on how much culture we use for the mobilization and how much we are going to try to scale up. It's different. Issue is some sort always been there for scaling of some sort of microbes that may be challenging for us. Uh, but maybe if we constant design a bioreactor with a constant concentration of microbes to encapsulate it, then that maybe you need not too much scaling up is required from a biological system. In the system, you already scaled, you can say, use a number of culture in reactor base. So it's limit the challenges as compared to others.
Cool. Thank you. I guess if there are no more questions, I do have one last question.
Excuse,
Oh,
can
yeah,
I
go ahead. Yeah, go ahead.
can I ask a question? Hello, good day. I'm John, and I am Miss Dr. Makauyag's friend, and it's a very interesting project. Are you? And I want to ask if you're concerned with having two cells inside. You have co-cultured with two yeast, right? Are you concerned with having both of them inside, immobilized inside that vesicle?
Which type of strain? Bacillus?
Yeah, you co-cultured with two kinds of yeast, right?
Yeah.
Are you concerned that should it have two of the different kinds inside, or is it okay that not all of them are inside, not the same thing are inside?
No, it's depend because if you try to encapsulate two different some type of microbe is uncompatible with one another. For example, I'm using co-culture, but one culture is affecting growth for the another culture. Then we try to separate it into encapsulate them into another polymers chain, other strain, another polymer. Then maybe these strain become compatible. But some microbes are
Mm,
already in compatible form. So then we use them in a mixed culture. Basically, when we use in a mixed culture in a
mm,
single beads, their interaction become more close as compared to separated one. Therefore, in close interaction, give
mm,
higher communication between these microbes from targeted molecule production as compared to others.
okay. Thank you. And is the particle size important or not really?
Yeah, it's important to it's term of we can say that many that's mean currently we just our target is sustain their production and yeah you can say that
Mm,
using reuse own control whenever you stop the reaction just separate the bees
mm,
from the reactor. So these are the application of these things. But if generally we should think that you can also design a pathways using this type of system. So for example, you use a 3 microbes, 4 microbes, yeah.
mm,
So that's up to you that how much you can utilize that to design a complete pathway for your targeted product, which may be not possible in natural way.
mm. Thank you.
Yeah, welcome.
Great. Any more questions for Haneef? If not, then I want to thank you again, Haneef, for spending the time to tell us a bit more about this kind of immobilization enzyme production.
Thank you.
So I guess we'll now move on to a more open roundtable discussion, see what we have, what anyone wants to bring to the table, like update each other about what's been going on so far. Any takers who want to go to start off? Good, it's always good when I have so many willing voices. So I guess I'm going to start off.
You became muted, Ian King.
Yeah.
Right, okay, sorry about that. Hi. So I don't know where I started muting myself, but okay, so the thing I'll bring to the table is that the Open Bioeconomy Lab, we brought to the meeting 2 meetings ago about the Open DNA Toolkits extension wish list. So this is, so we currently have, I think it was 20K pounds to spend on DNA synthesis to expand the DNA diagnostic collections we have, the open enzyme collection, open reporters, and so on and so forth. So I've put into chat the document, that this wish list document, and this wish list will be open for another couple of weeks to the end of May. So if you have any suggestions of what you want to have synthesized to add into these collections, then feel free to add to that list. So that's one update. Should I hold off and see if there are any willing participants to update a bit more about what they're doing recently? Whilst you're
Well, okay, I'll just maybe plug our program, what we are doing. So in IPRDC, we are working on the local production of enzymes and upscaling of it, and the creation of home brewed purification resins and bioreactors. So if you are interested in kind of exchanging notes or joining, because the engineering projects, they have their own kind of community there. So if you have some friends, or some engineers who are also trying to make their own DIY bioreactors or purification reagents, you can contact me and I'll connect you to them.
So yeah, do feel free to contact Aaron, but I've also posted in chat as well the Recon forum. So if you haven't joined that already, you can also log on to that and post any questions, queries, and then hopefully the community will gather around and help respond to any queries you might have. And I believe I flagged this up earlier in the meeting. To those who joined a bit later, we're currently curating members of the Recon community. So again, put in chat. This is a map that we've started working on. So it'd be great for you guys to put a pin in the map from where you are, where you're working. You could talk about what your expertise is in, but also what you're interested in finding about a little bit more about that you would also want to learn a bit more about. And this allows other members within the community to just have a look at the map, see if their expertise matches yours and vice versa. So I'm just throwing a lot of links in chat for you guys to have a look at. Is there anyone else who would like to tell us a bit more about their research in the last month or so?
Yeah, maybe I'll introduce my friend. So John Benson Joyce, my previous lab mate in Osaka University. He just graduated. He just got his PhD. Yeah, and I don't know if he probably gonna work with us in the future. I don't know. So yeah, if you want to say anything, John, feel free.
I'm just saying hi. Nice to meet everyone.
Lovely to meet you too, John, and congratulations. What kind
Thank
of
you.
what was your research in? Do you want to tell us a bit about that?
Well, my research was increasing the production of recombinant IgA on CHO cells. And to increase this, I did tested 5 different disaccharides and to see which one would increase the production more. So yeah, and I did also like analyze the glycosylations and the quality attributes such as aggregation. Yes, and that was my whole research.
Anyone here work with CHO cells, mammalian cells? Because I hear they're notoriously difficult to work with.
Yeah, they're very easy to contaminate. Yeah.
I think Paul worked with antibodies before, right? Are you there?
Uh, it's just E. coli.
Mm,
We're um, production of recombinant rabies protein for to be
uh,
used as antibody. Yes.
Seems like we have a mix of people in accordance. I'm working with mammalian. We've obviously Haneef, you talked about the yeast work. I think Brian, you also mentioned you worked with yeast. I mean, I'm a bacterial girl. I've always ever worked with E. coli.
No, I also work on bacteria. That's Bacillus species that's commercially basically very important for enzyme manufacturing. So my most work is you see the pollen Bacillus and bacterial strands. So and currently portfolio this is because these are the engineered strands that we use for we will just scale off that on you can say the compartmentalization this project. So my main work is currently I'm whatever enzyme production is only on Bacillus species.
Yeah, it's good
Yeah,
to know
hi,
them.
Yordi. Yeah, yes. Actually, yeah, wanting to know your email because previously I was not able to catch it, but okay, I'll email you and forward you to them, Yordi.
So was there a question that you previously wanted to ask Yordi?
Actually, her contact number because I think the window just closed immediately when I was about to get it.
Well, as I said, I can either if you're happy, we could always pass your contact to Aaron, but also direct you guys all to the forum, where you can have discussions as well if you prefer.
I think the engineering team of our project now is yeah, actually having regular meetings with the open bioreactor community actually. But they're busy recently, but yeah, I'll forward you to them. Maybe you can join some of their meetings. Oh,
Interesting. Thanks. I've just seen in chat. Oh, is there a link for open bioreactor
Oh.
meeting?
okay. It seems there are some people interested in the open bioreactor community. Okay, maybe I'll forward you to them. I do not know actually the schedule of their meetings, but what I know is they are doing that regularly right now. I'll forward you to them. So Jordi and Brian, forward you to them. Okay. Welcome.
Sorry, I was just trying to find links. So Brian, I've just posted into chat the GOSH forum, because I believe Tien from IPRDC, she ran kind of an open hardware, open bioreactor kind of FPLC, open FPLC meet and greets, I think a month ago, in March actually, I believe. So if she has a bit of a time and she's running that again, it'll be good to introduce you to that group. Cool. Well, I think I've kind of pushed a couple of you guys to say hi and to introduce yourselves a bit more today. Apologies. I mean, I didn't go down the route to directly target people, but it's great to hear the different kind of researches that everyone is doing around the community. So do feel free to message, chat in the forum, and so on and so forth. For, as I said, we have these monthly community meetings as well. So if there are no more updates for today, I will let you guys get on with the rest of your days. But I want to thank you all for joining us, and again, thank you, Haneef, for presenting to us today. Great. Well,
Thank you.
Thank you guys.
thanks for
Bye.
joining, and see you guys next time. Bye.
Thank you.